When severe psychological distress causes a person's sense of self to collapse, clinical psychiatry operates on a default assumption. We observe the ego dissolving, and we classify the event as a mechanical failure, a pathology occurring within the brain. Because we perceive this dissolution as a disease, the standard medical response is immediate suppression. The objective is to quiet the neural pathways, halt the mental breakdown, and force the patient's mind back into its previous organizational state. A divergent theoretical framework, however, proposes a different mechanism. The transformation program hypothesis argues that we have misidentified the process entirely. This model suggests that psychological crisis is an endogenous, biologically structured sequence. When an individual's existing self-structure becomes inadequate to meet reality, the organism initiates a phase of dissolution, dismantling the old architecture to prepare for a necessary reconstitution. If this hypothesis is accurate, a psychological breakdown is an adaptive program. By chemically intervening to stop the collapse, current medical protocols may be actively aborting the cure. To understand how an organism survives the deliberate destruction of its own architecture, we look at the metamorphosis of holometabolous insects. Inside the chrysalis, a caterpillar does not gradually sprout wings, it undergoes structural dissolution. The physical and neural matter of the insect breaks down into a largely undifferentiated biological soup, reorganizing around clusters of cells called imaginal discs. This presents a biological paradox. If the creature's entire neural network is liquefied, what happens to the specific lived experiences the caterpillar lived through before it entered the chrysalis? Recent empirical data from entomology indicates that experiential memory survives the total breakdown of the organism's physical structure and can transmit across generations, challenging our understanding of how history is stored in the body. The chrysalis appears to be a biological crucible where relational experience is carried forward even as the host form is destroyed. The foundational evidence for this survival of memory comes from Joe Nagai's 2024 research on Lepidoptera, specifically Papilio swallowtail butterflies. Nagai established a conditioning protocol for the caterpillars, introducing a naturalistic stimulus, lavender oil, and pairing it with a carefully calibrated, ethically grounded aversive shock. Before entering the chrysalis, the caterpillars demonstrated associative learning. When presented with the scent of lavender in a controlled maze, they actively avoided it. The critical data point emerged post-metamorphosis. After undergoing complete cellular dissolution and reforming their neural architecture, approximately 70% of the resulting butterflies still exhibited avoidance of the lavender scent. The organism liquefied, yet the memory persisted. The specific relational encounter with the environment survived the physical destruction of the brain that originally processed it. Nagai then extended the study to track this conditioned aversion through the insect's lineage. The results showed that the F1 generation offspring exhibited the exact same rate of lavender avoidance as their parents. The trait continued downstream into the F2 generation. Critically, these offspring were born with this behavior. They were never exposed to the lavender oil or the shock. This indicates a mechanism of epigenetic heritability. The original experience left a biological trace in a substrate that survived a chrysalis phase and passed directly into the germ cells. The organism's lived history encodes an epigenetic signature that bypasses physical destruction to shape the responses of its descendants. This data aligns with Dennis Noble's extended evolutionary synthesis, which views DNA as a read-only database rather than a rigid controller. Noble's principle of biological relativity states that cells activate specific genes in response to the environment. This allows for circular causation. The environment shapes the organism, and that shape is then passed forward. We model this using the recognition field equation. Gamma represents the system's capacity for recognition. In Nagai's experiment, the parent's encounter with lavender modified its gamma. That modified sensitivity transmitted forward, meaning offspring were born already tuned to avoid that stimulus. Nagai's data provides a biological value for the gamma parameter. It shows that recognition capacity isn't a static trait, but one actively shaped by history and transmitted as a modified field of sensitivity. This implies that the emergence of consciousness and the transmission of memory are linked by the same formal architecture, a process of recognition that carries forward through structural dissolution. We can apply this biological and mathematical architecture to human psychiatric crisis. Phenomenological evidence from isolated cultures shows a consistent sequence of ego dissolution, symbolic death, and reconstitution. They view this as an ancient biological phase. Modern non-suppressive models, like Finland's open dialogue, report functional recovery rates exceeding 80% by allowing this process to unfold. Similarly, psychedelic research indicates that therapeutic benefit is often tied to the intensity of the ego dissolution experience. By treating dissolution as an illness to be suppressed, conventional psychiatry induces a state known as iatrogenic liminality. Medication can be used to stop the dissolving psychological structure, artificially halting the metamorphosis to maintain the patient's original organizational state. The patient survives the intervention, but the biological sequence is arrested. They are caught between forms, a state of incompleteness that the medical system then manages as a chronic condition. The implications of arresting this transformation extend beyond the individual. Human studies, such as those of the Dutch hunger-winter cohorts, prove that environmental stress physically alters the epigenetic expression of offspring. These studies demonstrate that maternal nutritional stress during pregnancy produces heritable metabolic and psychological effects in children and grandchildren. If epigenetic inheritance operates as a bidirectional ratchet, both trauma and healing leave biological traces. If arrested transformation encodes transmissible stress markers, then a completed transformation, supported by genuine containment, may encode a biological signature characterized by a modified capacity for future transformation. The psychiatric encounter carries weight across generations. The system has the potential to produce either transmissible harm through suppression or heritable resilience through completed transformation. The solution lies in the first variable of the recognition field equation. The parameter G represents the boundary conditions and containment that hold a space during structural dissolution. True therapeutic intervention requires providing this containment, holding the human chrysalis so the organism can follow its own completion trajectory. The clinical imperative is to stop intervening to abort metamorphosis under the guise of symptom management. The goal of modern psychiatry cannot be to suppress the collapse of the old structure, but rather to build the containers that allow the human organism to rebuild, transform, and fly.